So here we are with chapter 11 in oxidative phosphorylation. So we know that energy conversion processes at the molecular level really sustain life on Earth, right? It's really been said previously that electron carriers that are produced, and go back to remember, the citrate cycle, glycolysis, all of those that lead to the production of energy through oxidative phosphorylation. This is how that energy payoff that we talked about before works out. We talked about the citrate cycle and glycolysis making electron carriers, but we haven't said, okay, well, they're electron carriers, so what? This is the so what, all right? So we're going to talk extensively about it. We had a brief thing last semester, so we're gonna do the detailed version now. So the chemiosmotic theory is really the beginnings of how we started to explain that payoff. And it was proposed in the 1960s, and it's one of the great unifying principles in biology, and I would really say in biochemistry. But it was really radical at the time, really, really radical. And really, it describes energy conversion in every single organism, which is pretty huge. And so it says that the cell is going to create a proton gradient, which we know is that H plus, which harbors potential energy. So it's like holding a ball up, right? And you drop it and it turns that potential energy into kinetic energy. Well, here we're gonna holding the ball up is like having the proton gradient. And then releasing the ball is allowing the protons to flow down their concentration gradient. And that's what we're going to use to drive ATP production. So, of course, we're going to talk all about the mitochondria. Yay, right? So, we talked about that, making that proton gradient, and it's established in the inner membrane space of the mitochondria. And so, chemiosmosis. So it's the movement of chemicals, right? The movement of protons down their concentration gradient. And as those protons flow, they're going to power the ATP synthase complex. Because we know if anything goes from high concentration to low concentration, that's simple diffusion. And we can use the energy stored in that diffusion process to make ATP. So the ATP synthase complex is bound inside the mitochondria in the same way that the electron transport chain is. And so we'll talk about that in lots of detail. But we know that this is an insanely highly conserved process. And the same process that's used in eukaryotes is used all the way down to prokaryotic bacteria, right? So that's what tells us it's very, very highly conserved. And chemiosmosis is also used in plants to synthesize ATP. It's through the thylakoid membrane instead of the mitochondria, but we're not really going to cover that. So we kind of talked about this, but let's talk in detail. We know that glycolysis and citrate don't produce very much ATP, right? They each produce 2 ATP. So if from one molecule of glucose, I know that I can get 32 ATP. Well, so far I've only generated 4. Well, that's only 12% of the ATP that I could make from one molecule of glucose, from the oxidation of one glucose molecule. It's only 12%. Where the rest of the 88% coming from? And they're stored, that energy is stored in the electron carrying molecules, and we'll see how they generate energy. So in order for that to happen, we have to make our electron carriers. And remember, our electron carriers, those are the things like NADH and FADH2, right? Those have to be oxidized in the electron transport chain in order to get the rest of that energy. So if we look at how this works, right? The whole point of the electron carrier molecule is to take whatever molecule has those electrons and we'll be in the matrix. This yellow area down here is the matrix. And so I'm going to have some electron carrying molecules, and I'm just going to represent them as electrons. That's it. And we know we're in the mitochondria, so we know we also have oxygen present. So when those electron carrying molecules get to the electron transport chain, they drop off their electrons. And in the process of dropping off electrons, they pump protons that were in the inner mitochondrial matrix and pump them to the inner membrane space. So we start to pile up protons here in this space until it gets to be a really high concentration of protons, right? So we have a higher concentration of protons in that inner membrane space and a lower concentration in the mitochondrial matrix. So we're establishing that proton gradient. So this is our source of electrochemical energy. And so what happens is that these protons, once there's a higher concentration in the inner membrane space, they're going to flow down their concentration gradient. But they can't do it by simple diffusion because there's a membrane there and it's charged. So it's not going to get through by simple diffusion. So the ATP synthase protein has a channel that those protons can go through. And as they go through the ATP synthase complex and come back into the inner mitochondrial matrix, at the same time, that is the cause for the change of ADP plus an inorganic phosphate into ATP. So we have now made ATP. And so we'll talk about this in a lot more detail. This is pretty simple, but it's a good little introduction. Okay, so we have to understand the electron transport chain. And you have to remember that what are we doing? We started out with glucose and we're going to oxidize it. So the whole time, anything that has energy, we want to oxidize it. So we're always going to be talking about redox reactions. And these redox reactions occur sequentially. Remember, we looked at that table of all of our standard reduction potentials, right? And so we know that NAD plus going to NADH, this is our strongest reductant, right? So this is going to pick up those electrons pretty easily and keep passing them and passing them and passing them, right? You need to have a higher standard standard reduction potential in order to transfer those electrons. And it's this difference between these different molecules, and these are not all the molecules we're going to talk about, but the difference in those reduction potentials provides the free energy for ATP synthesis, right? Because as those electrons move, they actually move those protons as well. Okay. So, and we know that when we're in the mitochondria, we have O2. Why is O2 so important? Because it is the strongest oxidant. This is why we need to do this in the mitochondria. So if we kind of look at this in just a little bit more detail, we have to think about the mitochondria and how it's behaving sort of like an electrical circuit. And you might not be familiar with electrical circuits, but electrical circuits have multiple parts. And the three kind of big parts are the resistor, the capacitor, and the battery. So a resistor is something that performs work. Well, what do I want to get out of the mitochondria? What's it need to produce? It needs to make ATP. So the work is going to be make ATP. The capacitor temporarily stores the energy that comes from the battery. So where does that energy go, right, while things are working? Well, it gets stored in here in all of the protons that are in this inner membrane space. So that's our capacitor, our proton gradient. And then what actually has the energy is the electron transport system. That's what provides us the energy. So what's really cool about this, so let me kind of zoom in, right? Okay. So when we look at this model, right, we know that this is our, oops, let me kind of do this. Okay, this is our electron transport complex. And it's really, really simple right now, but that's okay. And we're going to take our electrons and we are going to drop them off. And at the same time, we're going to take protons and we're going to move them into the inner membrane space. That's what we talked about. And then those protons, once they pile up, are going to flow through ATP synthase complex. This is what we kind of just showed. Well, what you have to kind of think about with this is because these are protons, you're going to have a difference in both the chemical gradient, which is the pH, difference in pH, and the electrical gradient. And we call that a membrane potential, right? So if I have more things in my, if I have more, if I have more protons in this area than in this area, then this is going to be more positive than this. And so that's our electrical, our membrane potential. We have a difference in the charge. It's still protons on one side and protons on the other side, but it's a difference in the number of them that gives you that. Okay, so is it always coupled like this? No. So sometimes you have protons that come in, right? They enter the inner membrane space, but instead of going to the ATP synthase complex, they have a pore that usually it's closed, but sometimes it can open up and our protons can flow through that pore and bypass the ATP synthase complex. That's the uncoupling. So when it's uncoupled, it bypasses the ATP synthase complex. So ATP synthase doesn't occur. Sounds like that's a bad thing, but it's really not. And the reason that it's not bad here is that sometimes it's more important to generate heat than it is to make ATP. So if we do this bypass, we call this protein channel and uncoupler. So our electron carrying molecules come in, drop off their electrons, our protons move from one side to the other side, and they don't go through the ATP synthase complex. They're actually going to go through this uncoupler, and that produces heat. So most of the energy is actually lost to heat, and that's actually good because you're going to see a lot of this uncoupler being expressed in brown adipose tissue, which is really, really important in thermoregulation for lots of metabolic processes. So it can actually be really good. The other thing that can happen is that if this ATP synthase complex is blocked and there is no uncoupler, then eventually you will have so many protons that pile up, you won't have anything here. And so NADH won't be able to be oxidized because there's no protons to pump. And so it basically gets overloaded and eventually the cell dies. And so this oligomyosin, I don't know if you recognize the name, but this is an antibiotic because it will bind to the bacterial ATP synthase complex, but not the eukaryotic. So we can take it and kill the bacteria and not the eukaryotes, us, which is great, right? All right. So how many mitochondria do you have in one cell, and how much of the electron transport chain and the ATP synthase complex are there? You don't have to memorize this, but it's just kind of cool. Usually the standard person has like 10 to 100 mitochondria per cell. And if you look at the surface area, you'll have about 5,000 micrometers squared per cell of mitochondrial membrane, right? This is our inner mitochondrial membrane, right? So it's this one that can be folded into all the cristae and that kind of thing, right? So that's our inner mitochondrial membrane. So if you take that surface area and you multiply that times the number of cells that we have, okay, we have, like a trillion cells. That gives you 185. Oh, did I not? Yeah, 185 meters squared. So that's 26 full-size soccer fields of, I love numbers like this, of inner mitochondrial membrane per person. Like, good Lord, that's a lot. That is extensive cell surface area, right? Not cell surface area, mitochondrial intermembrane surface area. So that's where all of these ATP synthase molecules and electron transport chain molecules are all embedded in this membrane. So you have a ton of them. And if you look at different animal species, so the more the muscle has to work, the difference in cellular composition, right? So dark meat is tissue that has to have a high mitochondrial content because it's going to be doing exercises that require oxygen. And so the dark meat is dark because you have lots of myoglobin there. But to support that, you also need to have a ton of mitochondria. And so you can always order, instead of dark meat, order the one with more mitochondria. Like, that's awesome. So that's our, sometimes they call that slow twitch muscle. The dark meat is slow twitch, right? And that's for like sustained energy, right? Fast twitch is where you have to, it was like the breast tissue, like if they had to fly. But you see a really big difference in commercial turkeys, like if you're at Thanksgiving or Christmas, because turkeys rarely fly. Whereas game meat, you don't see so much of a difference in the color of the tissue because, yeah, they got to fly and survive. Okay. So when we look at the components of the electron transport system, you need to know the names of them. So start getting familiar, but they're going to make sense as we go through and explain them. So there are 5 complexes that are required for oxidative phosphorylation. So what we need to know is, what are the complexes? Where do they get their electrons from? So the very first complex gets their electrons from NADH, and it is going to pump 4 protons after it picks up the electrons from NADH. And so we call it NADH because that's where the electrons start. Then it's going to eventually send it to ubiquinone. So that's why we name it NADH ubiquinone. And then what kind of reactions are happening here? These are oxidation reduction reactions. So we call it oxidoreductase. So then complex 2 picks up from our other electron carrier, FADH2, but it does not pump any protons. And you'll notice, you probably recognize this enzyme, succinate dehydrogenase, and we'll talk about that later. Then complex 1, the electrons come from 2 and come from 1 and get dropped off at 3. So it's the ubiquinone that carries those electrons from complex 1. So it's ubiquinone, and then we're going to send it to cytochrome C. And so that's why we have the names. The names tell you the pathway. And we have oxidation reduction reactions again. Then the electrons go from 3 to 4, and we pump, oh, and I didn't say that. In complex 3, we're going to pump 4 protons. And then in complex 5, we're going to pump another 2 protons, and we call it cytochrome C oxidase because, well, I'll get there. We'll get there. And then the last complex, we don't really call it complex 5 anymore. Really, we just call it ATP synthase complex. And so this is where we're no longer generating protons, we're no longer generating them, we're using them. For every 3 protons that flow through the ATP synthase complex, we're going to make 1 ATP. And I'll show you how all of that works. But what is really important to remember is that these are all transmembrane proteins. They all have to be embedded because we said they were embedded in this inner membrane of the mitochondria. So just remember that. But then the functional domains have to face towards the matrix because that's where all of these NADH molecules and FADH2 molecules are located. And so the functional sides have to be facing the matrix. So, and that's right. We'll talk about some questions kind of at the end. But so when you look at the electron transport chain, this is kind of the same thing, but it's broken down a little bit more, where you have complex 1, complex 2, complex 3, complex 4, and then this is really complex 5, but it's really the ATP synthase complex. And then we have some other transport proteins that we're going to talk about. But we're going to go in detail and we're going to talk about each of these individual steps and what's happening at each one. Okay, so that is the end of section 1.